Our speaker for today is an eminent scientist and inventor in neuroscience. Professor Ed Boyden is the Y. Everton Professor in Neurotechnology at MIT, an investigator of the Howard Hughes Medical Institute and the MIT McGovern Institute, and Professor of Brain and Cognitive Sciences, Media Arts and Sciences, and Biological Engineering at MIT. Prof. Boyden received his PhD in neuroscience from Stanford University as a Hertz Fellow, working in the labs of Jennifer Raymond and Richard Shen, where he worked on the molecular mechanisms of memory storage. In parallel to his PhD, as an independent side project, he co-invented the optogenetic control of neurons, which is now used throughout neuroscience. Prof. Boyden has received numerous awards for his immense contribution to neuroscience and bioengineering, including the Breakthrough Prize in Life Sciences 2016, the Croonian Medal 2019, and the William Exner Medal 2020. He now leads the Synthetic Neurobiology Group, which develops tools for analyzing and repairing complex biological systems, such as the brain. These technologies include expansion microscopy and optogenetic tools, which he will share during his talk entitled Tools for Analyzing and Repairing Biological Systems. Professor Boyden, please.
Thank you. It's great to be here and to talk to you a bit about the technologies we're developing for understanding and controlling complex biological systems. Personally, what motivates me is this question of, can we understand the brain? And in particular, how the brain generates things like thoughts and feelings. Can we get to the point of actually computationally modeling the brain? And one of the key reasons that this is hard is that the complexity of the brain is enormous. The cubic millimeter of the brain contains 100,000 cells connected by 1 billion connections. And each of those connections and each of those cells is operating with millisecond precision, millisecond precise electrical pulses within cells, and millisecond precise chemical exchanges between cells. So my group, we've been working on developing, and now we're working on applying, 3 kinds of technologies: ways of mapping molecules throughout cells and cells throughout tissues, ways of controlling and observing the high-speed dynamics of these cells and tissues. And I would argue that these tools are now finding applications not just in neuroscience, but throughout all of biology, because lots of biological problems relate to complex systems. The immune system is a body-distributed system. A metastatic cancer will wander through the body. If you care about aging or development, these are all complex systems made of many cell types, made out of many different kinds of molecules. So as you'll see, we're finding lots of applications of these tools in areas ranging from virology, very appropriate for the current time, to cancer biology, to developmental biology, and the list goes on and on. Now, for each of these 3 problem spaces, we had to rethink the problem from first principles, because the spatial and temporal complexity of these systems is very, very broad. Think about a brain cell. A human brain cell can be centimeters in spatial extent, by far the largest cells in the human body. But the connections between cells are nanoscale, and the products of genes are also nanoscale, proteins, for example. So if you want to image a brain with molecular precision, you have to cross many spatial scales, from the nanoscale to the macroscale. And so in the first half of the talk, I'll tell you about expansion microscopy, a technology we've developed which allows us to cross those spatial scales from nano to macro. In the last half of the talk, I'll talk about crossing timescales. So of course, in biology, lots of complex problems happen over days to months to even years. But the fastest physiological events in the body, like electrical pulses within brain cells or other signaling cascades, those happen with millisecond precision. So sometimes I say our job in our group is to figure out how to cross space and time. And so I'll tell you 1 story about space and 2 about time today. Let's start with space. So many biological problems relate to the mapping of molecules throughout cells and cells throughout tissues and tissues throughout entire organs and even organisms. Now, many pioneers have developed nanoscale imaging techniques in the past, but they tend to be difficult to apply to large 3D objects, especially if you want molecular information. With X-ray and electron microscopy, it's possible to see very fine-scaled things, but it's still very difficult to apply these techniques to large-scale 3-dimensional objects, like brain circuits or intact tissues from a cancer patient, let's say. At the other side, we also have seen great progress in super-resolution microscopy, like STORM, PALM, and STED in recent years. But these also struggle to image large 3D objects. So in my group, about 5 years ago, we announced sort of a surprising discovery. In contrast to 300 years of zooming in with a lens to take a picture or magnify an image for your eye, we decided, what if we could physically magnify a biological specimen itself? What if you could weave chemically a dense spiderweb-like mesh of swellable polymer so that it goes inside cells, outside cells, in between biomolecules, around biomolecules, and that dense swellable mesh of polymer will then allow us to expand the biological system. So suppose a swellable polymer is like the stuff in baby diapers, these highly charged polymers, such as sodium polyacrylate. Well, what happens when you add water to sodium polyacrylate? Here's some kind of analysis that was being done several decades ago by scientists like Toyoichi Tanaka in 1980. He was studying the physics of soluble polymers. You add water to sodium polyacrylate, the water is drawn in through osmotic force, and the polymers will swell apart from each other. And because they're highly charged, they repel, and you get these enormous volumetric increases in size, 1,000-fold increase in volume in a matter of minutes. And so Tanaka and colleagues studied the physics of this. What about getting the polymer inside the cells, in between the biomolecules? Well, around the same time, surprisingly, Peter Hausen and Christine Dreyer published a paper, 1981, where they were able to embed biological specimens in a hydrogel. They used an uncharged polymer, polyacrylamide, rather than a charged polymer, polyacrylate, but they were able to soak preserved tissues in a solution of monomers, and the monomers would then self-assemble into the polymers. So they added acrylamide, and it would self-assemble into polyacrylamide. And they were then able to use this strategy to facilitate staining and imaging. So if we could weave a dense spiderweb-like mesh of soluble polymer, anchor biomolecules to it, and then add water to expand the biomolecules away from each other, you might imagine we could turn a cell like the one on the left to something like what you see on the right, a constellation of biomolecules hovering in space, but much, much bigger. They've been expanded away from each other. Two biomolecules that are touching are now some minimum distance apart, and two biomolecules that are some distance apart have now been separated by some linear scale factor. So the next few cartoons illustrate the chemistries we had to develop in order to make this occur. In this cartoon, proteins are shown in brown and handles are shown in purple. And we developed handles that will bind to different biomolecules like proteins, nucleic acids, and so forth, so that they are equipped with a little anchor. Then if we pull on the anchors, we should be able to pull the biomolecules apart from each other. Next, we have to weave that dense spiderweb-like mesh of soluble polymer. And we follow a recipe that's not too different from what Hausen and Dreyer published in the early 1980s. We add the monomers, shown as white spheres, and they self-assemble through a process called polymerization into long chains of swellable polymer. We use a charged monomer, sodium acrylate, and the polymer lines that you see here represent the polyacrylate. When the polyacrylate encounters the handle, they form a covalent bond. So now the polymer threads can generate force, and the handles can convey the force. Finally, we have to soften the specimen. We use enzymes, heat, and/or detergents. And then when we add water, the polymer threads fall apart from each other, but this time, the biomolecules, because of those handles, come along for the ride. Importantly, the polymer mesh starts out very, very small. In the upper left is a little cartoon. The spacing between polymer threads is just a few nanometers, about the size of a biomolecule. In the lower left is the polymer after it's swollen. And in panel B, at the top middle, is a piece of mouse brain. And panel C on the right is the same mouse brain tissue after we expand it, about a day and a half later. This is work that Fei Chen and Paul Tilburg and I published in 2015 when they were both grad students in my group. Here's a time lapse of a piece of mouse brain tissue being expanded. We formed the polymer earlier, and right there we add water. And I hope you can see the polymer embedded brain is now swelling before your very eyes. This time lapse is sped up by about 50-fold. So by design, this dense spiderweb-like mesh, the softening, the anchoring, these are all designed to make the expansion process as even or isotropic as possible. But this is biology, we have to prove it. And so we spent a lot of time in the early years taking pre-expansion images with a classical method that lets you see nano-resolution things like super-resolution microscopy. And then we would take a post-expansion image on a regular old microscope, the kind that you have at, ordinary core facilities at many universities. And the error that we introduce is not zero, but it's very, very small error, around a few percent over a microscope's field of view. We can also gauge the error by imaging things where we know the ground truth. So here are some microtubules in cells. They've been labeled with fluorescent antibodies that bind to tubulin, the protein inside the microtubule. And then we can take a picture on a microscope. We can take our picture, deconvolve it by the known ground truth, which we know from decades of electron microscopy and X-ray imaging. And from that, we can estimate the resolution. So in these images, so far in the talk, we're expanding by about 4.5-fold in each direction, about 100-fold in volume. 4.5-fold cubed is 100 approximately. And if we measure the effective resolution through the method I just mentioned, then we get an answer which is matching that of the 4.5-fold expansion. So if we're using a 300 nanometer resolution lens, 300 divided by 4.5 is around 70 nanometers, and that's what we get for the resolution. Now, as I told you earlier, the polymer has a spacing of around 2 nanometers or so. If we expand more, can we get more resolution? And as I'll show you later, the answer is yes. But there's a lot you can do with 4.5-fold expansion. On the left, from top to bottom, we're zooming in on a piece of mouse brain tissue. The color code is shown on the left. These are all fluorescent antibodies binding to specific proteins. And what you can see on the left, we're zooming in, is that eventually we get blurry because we're using a regular old microscope at our local core facility to take these pictures. In the right column, though, I hope you can see, after we expand, we get very, very high precision. And the color code, as shown on the left, is interesting because we're labeling synapses, the connections between brain cells. So bassoon in blue is a presynaptic protein. Homer 1A in magenta is a postsynaptic protein. And as you can see, we can resolve cleanly these pre- and postsynaptic sides of neural connections, as shown in the lower right. Furthermore, if we look at the distance between these pre- and postsynaptic protein densities, we get the same answer that was measured using super-resolution microscopy by Xiaowei Zhuang and Catherine Dulac, about 150 nanometers in the upper right-hand corner. But this time we took that image on a regular old confocal microscope. So one way to look at this is, we're expanding the tissue and making it bigger, but that means we get a bit of a best of both worlds effect. We're getting the speeds of classical imaging, but the resolutions of a nanoscale imaging method. So we can take images that have billions of voxels in 3D on regular old microscopes. As you can see here, from top to bottom, we can zoom in down to nanoscale domains within cells. Now, to drive the point home, another reason why this best of both worlds is so important is that expansion, as a byproduct of expansion, you get this cool effect where the tissue becomes clear. So in the upper left is a piece of brain before we expand it. In the lower left is the same piece of brain after we expand it. And if you think about it, expand 100 times in water, it's about 99% water. What that means is you can use cool tricks. So we worked with Eric Betzig, who himself won a Nobel Prize for his work on super-resolution microscopy. And originally, he was very skeptical of expansion. He thought it sounded like not a very good idea. But as we started to do experiments together, he realized that it offered some really powerful benefits. So here in the upper right, we are antibody staining against mitochondrial and lysosomal proteins. At the bottom, we're antibody staining against myelin proteins in the brain. And if you expand, you can use a clever trick called light sheet imaging. Shine light from one angle and take a picture from a different angle. And what we found working with Eric is that if you do this, you can go orders of magnitude faster than the nearest competing nanoscale imaging method. So in this paper in 2019, spearheaded by Rei Gao, Sho Asano, and Gokul Apadiula, we showed that we could go almost 1,000 times faster than the nearest competition. And now we think we can see a path to maybe getting to, who knows, tens of thousands of times faster, maybe even 100,000 times faster than classical nanoscale resolution imaging methods. Again, speed gives you scale. So here's the thickness of the mouse cortex, layer 1 on the right, layer 6 on the left, and each image is a zoom in of the dotted box above, and same color code as before. And as you can see, we can get very, very accurate images across an extended spatial scale. Now, one of the cool things you can do, of course, is to look at proteins like we showed you earlier, and this is turning out to be super exciting. But you can also look at the wiring of the brain. A group at Harvard developed a technique that they call Brainbow, where you can randomly express fluorescent proteins in neurons through delivering cocktails of viruses. That's what you see at the bottom here in the mouse brain. At the top middle, though, you can see that if you zoom in, you quickly hit the resolution limit of a typical microscope. Like we see with these arrowheads, green blobs, what are they? But in the upper right, you can cleanly resolve after expansion that the green blobs are not green blobs. We can resolve clean pathways, individual axons, and therefore look at the wiring of the brain. So to summarize the first part of the talk, we've discovered that we can evenly expand preserved biological objects through a chemical process. This converts existing microscopes into nanoimaging devices, and they can be used to visualize a wide variety of proteins as well as anatomical structures, thus democratizing nanoimaging. I should point out that all the protocols are on our website, expansionmicroscopy.org. Over 160 papers and preprints have already come out doing some kind of expansion protocol. And we've done hands-on training of hundreds of groups on how to do the experiments, although the most recent protocols papers, I think, explain the step-by-step procedures with enough detail that you don't need to come in person to see us do it, which is, of course, very important with our modern pandemic world. Okay, everything I've shown you so far is with protein visualization, but when Oswasi did his PhD in our group, he and Faye co-spearheaded a project to anchor and expand multiple kinds of biomolecules away from each other. So here we're applying a protein anchor and an RNA anchor, and we're expanding both away from each other. The proteins, well, some of them are visualizable here because we have yellow fluorescent proteins shown in green, and then the purple dots are post-expansion fluorescent in situ hybridization, where we are labeling RNAs with fluorescent probes after we expand. This is very important because now we can look at the location of nucleic acids as well as proteins in the same sample. How big can we expand? Well, everything I've shown you so far is expand roughly 4-fold or so. When J.B. Chang was in the group, and now Deblina Sarkar, Oswasi, Jinyun Kang, and Margaret Schroeder have been working on improved versions of this. So if you want to try this out, don't use the published version. We have a new version which uses all off-the-shelf chemicals. We can take a piece of cells or tissue, polymerize and expand it, form a second polymer in the space opened by the first, and expand it again. So it's exponential amplification with a linear number of steps, but it's spatial amplification. One thing you can see with the most advanced, the highest resolution, super resolution methods like STORM is the sidewalls of microtubules, which of course should look tubular. At the top in panels A, B, and C, you can see data collected by STORM microscopy by Xiaowei Zhuang's group, who kindly helped get the data. In D, E, and F, you can see that we can also use double expansion to visualize microtubules. Synapses, I showed you Bassoon and Homer in the upper left earlier. They looked like they were touching, but with double expansion, you can see the synaptic cleft in between, and this is an excitatory synapse. You can see glutamate receptors. In panel D in the lower left, you can see inhibitory synapse with GABA receptors in the synaptic cleft. And here's Brainbow again, where you randomly express fluorescent proteins, unexpanded in panel A, expanded once in panel B, and in panel C is doubly expanded. So there are many things we don't yet know. Since we validated this by comparing our images on regular microscopes to classical super resolution methods, we can't validate better than their resolution. And so there are a lot of questions, like what about down to single-digit nanoscale dimensions? Well, since no microscopy technique by itself can get down to that level, we need to think of new ways to validate under the 10 to 20 nanometer range. Nevertheless, there are lots of things you can do. when Yongxin Zhao and Octavia Bucur were working with my group, Astrid Wines, Stu Schnitt, and Andy Beck's groups, we showed that we could apply this to human tissues. This is a crowded slide, but on the left are normal, and on the right are cancer-containing human tissues. The color code is in the upper right, DAPI in blue, and antibodies against vimentin and keratin 19 in green and red. And from top to bottom, we see examples of human prostate, lung, breast, pancreas, and the list goes on and on. So there are lots of examples now where people are visualizing nanoscale domains in disease states. In panel D, you can see a kidney biopsy from a human patient. You can see these finger-like processes in this electron microscope image. In panel F, there are no finger-like processes because this patient had a nephrotic kidney disease. Panels E and G are from the same patients, respectively, as D and F, and with expansion microscopy plus antibody staining, the color code is in the upper left, we can visualize these fine processes. So what required electron microscopy before can now be done on microscopes that pretty much everybody at their core facilities has some kind of access to. We did a breast cancer study. Many diseases, if you can detect them earlier, you could save more lives. But early in a disease, it's harder to detect these conditions. So we hypothesized, could we expand these biopsies and turn the small, invisible changes early in a disease to these big, obvious changes? And that's what we found. So we worked with a team of pathologists to study early breast cancer, a stage where doctors disagree up to half the time. And we found that after we expanded these biopsies, we could train a machine learning algorithm to better classify these specimens. Limor Freifeld, when she was a postdoc in the group, we looked at the zebrafish brain, a very popular organism in biology, and showed that we could expand zebrafish brains and visualize, as you can see here, synaptic connections between different kinds of neurons of importance for the fish behavior. Jay Yu just finished up his PhD in our group, also showed that we could expand C. elegans, another very important model organism in biology. And this slide really isn't meant to be read. It's just full of text. But the point I want to make is that expansion microscopy is out there and people are using it. As I mentioned earlier, over 160 preprints and papers have already come out doing some kind of expansion process. It's being used to study how the flu virus spreads, how microbes are organized within, how fruit flies sense mechanical inputs, how phase-separated parts of cells like stress granules operate, how molecular motors are distributed, and the list goes on and on and on. Studies of the microbiome, studies of plants, studies of receptors, studies of brain tissue removed from the operating room from a neurosurgical patient. So to summarize the first half of the talk, we've discovered we can evenly expand biological systems in a process called expansion microscopy. We've trained hundreds of groups on how to do this, and many, many groups are using it to do real science. It's a very practical way of doing nanoimaging because it's inexpensive and you don't need any advanced skills or advanced equipment. And it's being applied in a wide variety of biological and medical domains. In the last half of the talk, I want to address the complementary arena of time. So of course, expansion microscopy won't work on a living thing. It only works on preserved specimens, non-living things. And so in the last part of the talk, I want to talk about a few areas that we worked on for controlling and imaging high-speed physiology in living systems. The first of these I'll talk about is optogenetics. Can we control high-speed cellular signaling using light? And in particular, we started with neural activity. Now, neural activity is electrical in nature, and we wanted to control electrical activity because if you could drive neurons, you could figure out how they trigger behaviors or pathology conditions, or maybe repair the brain. If you could silence neural activity, you could figure out what those neurons are needed for. So when Carl Dijsselroth and I were both students, we started brainstorming about just going through all the laws of physics. How can we deliver energy to the brain? It could be light, it could be mechanical force, it could be magnetic fields. And we decided light would be the best if we could do it because light, can be aimed and it's fast. The next question is, do you make a light sensor or do you find them? And it turns out that all over the tree of life, you can find in single-celled microbes light-sensitive molecules. 7 transmembrane proteins, as shown in the lower left, that convert light into an ionic gradient. So you put the gene that encodes for this protein into a cell, we thought. If the cell can manufacture the protein, it's like installing a little solar panel. You put a molecule into the membrane that converts light into electrical energy. So the first of these to be found, reported in 1971, was a light-driven proton pump, shown in structure form in the lower left. It binds a chemical partner called all-trans-retinal, and it's found in microbes, such as you can find in salty water, like in the upper left. A decade later, people found light-driven chloride pumps from the same species of microbes. And around the turn of the millennium, several groups found light-driven ion channels from different microbes, single-celled algae. Flash forward to the current day. What we found is that members of all 3 of these classes can be discovered that are safe, fast, and effective enough to work in delicate, high-speed brain cells. Each of these molecules is genetically encoded. We put the gene into the cell using a virus or other transgenic method. We shine light of a certain color, shown here in little cartoons, green, yellow, and blue, for example, and that light will then cause the protein to act. Importantly, the all-trans-retinal that I mentioned, we found just out of sheer serendipity, is present in neurons in the mammalian brain. So that's just a bit of luck. Didn't have to happen that way. And more serendipity, we found that these molecules were safe, fast, and effective enough in neurons. So on the left-hand side, when Brian Chow and Xue Han were working with me, we found light-driven proton pumps that we could express in neurons. When you shine green or yellow light, they pump protons out and silence the neuron. In the middle column, Xue and Amy Chuang, when they were working with me, found that we could put a light-driven chloride pump into a neuron and shine yellow light, or as I'll show you later, even red light on it and silence the neuron. So the left 2 columns allow us to shut down neural activity to find out what that neuron is necessary for. And the right-hand side, originally collaborating with Carl, and then later with Nathan Klepecki, Youngkoo Cho, Amy Yang, and Damien Park working with me, we found that we could take light-driven ion channels and put them into neurons and use those to drive neural activity with light. So in recent years, we've been trying to push these molecules to their physical limits. One limit is color. Blue, green, and yellow light, as we used with the older molecules, doesn't go very deep in the brain, but red light does, because red light is less absorbed by blood. So we've been, Amy Chuang, when she was a PhD student in the group, went through a molecular screening process and mutated a molecule further to find out that we could have a light-driven chloride pump that we nicknamed JAWS that we could put into the brain with a virus, and then we could shine red light even through the intact skull of an awake-behaving mouse and shut down neural activity several millimeters deep. So this could allow non-invasive methods for neural silencing. And later, Leah Acker, working between our group and Bob Desimont's group, we found that we could actually apply this to inactivate large volumes in the non-human primate cortex. When Nathan Klepecki was a grad student in the group, he worked on a similar quest for red light activators. And so we found a molecule that we called crimson, which can be used to activate neurons with red light. And that's proven very popular for the study of fruit flies, for example, where red light or even infrared light can be used to excite crimson neurons. And that means that you can activate neurons without causing behavioral artifacts. Fruit flies don't like visible light when it's flashed at them. We also pushed the speed limit. Can we make molecules or find molecules that are very fast? And through these screens, we found a molecule that allows very fast activation, which is important, for example, for studying things like the auditory system, which is very high speed. We call this molecule Chronos. Chronos and crimson, used together, it turns out, can be used to control 2 different pathways in the brain with 2 different colors of light. Put crimson in one set of cells and Chronos in a second set of cells, and then you can use red and blue light to differentially control them. But what about going beyond 2 colors? Well, it's difficult to have, 100 colors. So we worked with Valentina Emiliani in Paris to develop a way to target light to individual neurons and excite them one by one. Valentina's group is expert at inventing holographic projectors for the brain. As you can see in the top schematics, these are microscope schematics, where you bounce laser light off of a spatial light modulator, and then focus the light through a lens onto the brain, and that allows you then to control a 3D set of cells. As you can see in the image at the bottom, Valentina's microscope can aim light at 6 different points. So a holographic projector basically makes a 3D sculpture of light in the brain. So Dimitri, Tanese, and Valeria Zampini in her group, and Noor Shemesh in my group, we teamed up and said, could we make a molecule that's ideal for both for the holographic projector strategy? And one thing we wanted to do was to figure out if we could express the molecule just at the cell body. On the left, imagine you aim light in the dotted circle. You'll still excite all 3 starred neurons because the axons and dendrites of 2 of the neurons pass through the dotted circle. But if you can express the molecules just so they end up on the soma, on the cell body, like on the middle column, then the dotted circle will not excite those axons and dendrites. This is an idea that was applied to optogenetic molecules by Frank Werblin and Zhao Pen and McLean Bolton and others. So we thought, could we devise a molecule that would be optimized for the holographic projection strategy? And Nathan, when he was in the group, had discovered this molecule that we call COCHR. If you express it, it ends up all over the cells, axons, and dendrites, like on the left. But in the next to left column, you can see that we can fuse it to a small peptide and localize it just to the cell body. In the upper right, you can see that allows us to be focal and just excite single cells, but not their neighbors. And COCHR is a very powerful molecule. So as you can see in the lower right, we can get very, very precise neural activity. So to summarize, we've discovered that these microbial opsins, natural molecules, can be used to mediate the optical control of neural activity. And we've now sent these molecules out to, at this point, probably thousands of research groups all over the world. It's being used to activate all sorts of cells in the brain to figure out how they trigger behavior, or to shut down cells in the brain and figure out what they're needed for in a behavior or pathological state. And it's even being used to discover patterns of brain activity that might have remedial potential for helping patients with Parkinson's, Alzheimer's, and so forth. Now, it's very important to point out that with optogenetics, as we call it, we were really, really lucky. These molecules, basically out of the box, had enough safety, speed, and efficacy to mediate optical control of neural activity. It was really serendipitous. What about the opposite, though? What about the imaging of neural activity? In this case, we have not been so lucky. The natural world has not evolved molecules that allow brain cells to broadcast their activity. So Erica Young and Kiro Pijakovic, when they were postdocs in my group, we decided if the natural world won't evolve these, let's evolve them ourselves. And so we built a robot that evolves proteins. Now, directed evolution has been around for a while. Frances Arnold, for example, recently won a Nobel Prize for her work on initiating the field. But we wondered, look, we need to screen in multiple dimensions at once: safety, speed, efficacy. If you evolve one direction but not others, you might actually make things worse for your goal. Secondly, we really wanted to do the evolution in mammalian cells. So, if you evolve something to work in bacteria, you might not evolve it to work better in the brain. So we devised a robotic screening strategy. How does directed evolution work? Well, take a gene and make millions of mutants. Some are better and some are worse for any given goal. We put those mutants into mammalian cells in cell culture in the top row of this cartoon. Then in the bottom row, we use a robotic microscope to scan the cells and then pick out the cells with a robotic arm that have the properties we want. They're safe, they're efficacious, and so on and so forth. We decided to start by trying to build a fluorescent voltage indicator. That is, a molecule that's genetically encoded. You'd express it with a virus in a cell. The cell would manufacture the protein, and the proteins would then allow the cell to sort of blink at you when the cells were active. Fluorescent, so we shine red light, and they might emit redder light, for example. And that's what we found we could do. We started with Quasar-2, a molecule from Adam Cohen's group, which was a candidate voltage indicator, and we evolved it into a molecule we call Archon. Then we made a somatic version of Archon to clean up the fluorescence, analogous to the somatic control that I told you about earlier. And then in a collaboration between Shuo Han's group at BU and our group, Seth Ben-Susan and Hua Sang in Shuo's group and Kiro Pijakovic in our group teamed up to express this somatic Archon using a virus in the brain and then we could image it with a microscope, as shown in the upper left. Shine red light from a laser on the brain, collect the fluorescent light on a camera, and what you can see here are lots of traces that look like they're picked up by electrodes, but they're not. They're actually being picked up on a camera. What that means is we can actually image neural activity. Here's a population of cells, and these are mice that, although they're head fixed under the microscope, they are awake and behaving. We can pick up the activity of populations of cells under the microscope. This is really exciting because now we can really start to analyze neural dynamics with the precision that we require. I should point out that this idea of focusing a molecule at the cell body has a lot of other potential applications. I'll just give you one that we recently published, led by Urshamesh, Chengyang Lingu, and Kiro Pijakovic in our group, along with many collaborators shown in the lower right. One of the most popular ways of neural imaging right now is to image calcium, because calcium goes up in a neuron when it's active. And so we wondered, could we actually take a calcium indicator, such as GCamp6, developed at Janelia Research Campus, fuse it to a peptide, and localize it to the cell body? And we did this collaborating with Amy Keating's group by fusing GCamp6 to a coiled-coil domain protein pair. And what we found was we actually could get these proteins, as you can see in panel C at the top middle, to be located at the cell body, whereas normally they go all over the place. And that means we can clean up the background fluorescence and get very precise activity from neurons. Here's an example, again, from the Han lab. If you look at panel B versus panel A, or panel D versus panel C, these are each showing a somatic calcium indicator shown next to a conventional one. And you can see there's a lot less background fluorescence when we target the cell body, and it cleans up the activity really well. We're getting a lot less artifactual information bleeding from one cell to the next. And that's what you see in panels J and K at the bottom. When we express these molecules just at the cell body, then we get less light from one cell artifactually being attributed to a neighboring cell. So I think this idea of molecular focusing, where we take a cell and target the parts of cells, take a molecule and target the parts of cells, is really powerful. And combined with this idea of robotic evolution, if we can make lots of new indicators, we might be able to create a whole family of these to look at a whole variety of different physiological functions. So to wrap up the third and final story, we built robots to evolve what the world would not evolve for us, and we're very excited now that we can make lots of new indicators of different biological functions. Just as with expansion microscopy, we distribute all these tools freely. Go to our website, synthneuro.org, synthneur.org, and you can get access to all these molecules from Adgene or other nonprofits. In-house, however, we're very excited about joining these tools together. Can we image activity throughout a brain circuit? Can we perturb activity using optogenetics? And then can we use expansion to make a map? And my hope is that these tools, although they're very powerful alone, can be combined to build a new kind of workflow for looking at neural computation. Maybe we can even start to make computational models of how neural circuits work to generate things like complex behaviors, or how they go wrong in brain diseases. So along the way, I think I've acknowledged all the people who have led the projects, but I want to acknowledge an even longer list of people, both current and alum members of the group at the top, and an even longer list of collaborators in the middle who helped make all this research reality. This is truly not just an interdisciplinary field, but an omnidisciplinary field. And we're very eager to collaborate or teach. So take a look at our website in the upper right-hand corner. We're eager to share all of our tools as freely as humanly possible, and we're very excited to help in any way. And with that, I'm happy to take questions.
Thank you very much, Professor Boyden, for your exciting talk about expansion microscopy and optogenetics, as well as fluorescent proteins. Now, I can't speak for everyone, but I'm sure this talk has definitely expanded my mind. We'll now open the Q&A session to the audience, and due to time constraint, we can only allow one question per person. Of course, more if there's more time. Kindly state your name before your question. So to get the ball rolling, I should just ask the first question. So when you are homogenizing your sample for expansion microscopy, how did you avoid destroying your sample? For instance, you mentioned that you use enzymes. How can you prevent the enzymes from breaking down the proteins which you're actually trying to study? And how do membranes actually expand? For instance, like membranes, they have a certain surface area. So if you expand it, the surface area will expand. And how exactly does it expand?
So we're expanding everything apart from each other. Anything anchored to the polymer is being expanded away from each other. So all the proteins are being pulled apart from each other. If lipids are anchored, they're being pulled apart from each other. It's like you draw a picture on a balloon and you blow it up. It's the same picture, but all the ink molecules are farther apart. So the way to think about it properly is that all the molecules are being expanded apart from each other to first order.
So in that sense, can I say that the phospholipids are actually further apart from each other after expansion?
If they're anchored to the polymer. I mean, if you don't anchor them to the polymer, they'll be washed out.
I see. Thank you. Are there any further questions?
Yeah, hi.
Hi, how are you?
I have a question about the tissues that you can use. You mentioned human brain. Can you use other things like muscle, for example?
Oh yeah. It's been applied to all sorts of stuff. It's been applied to spleen, pancreas, skin. The list goes on and on and on. It's been applied to C. elegans, zebrafish, plant tissues. It's been applied to bacteria. So if you want to expand something, a good way to start is to look up on Google Scholar, expand microscopy, press the thing you want to expand, and then you can see how they did it. But most people use a pretty standard protocol at this point. To expand bacteria, the group at Stanford, Bo Wong's group, added an extra enzyme to soften the cell walls. Most people are using the same basic protocol at this point.
I guess my point, I'm particularly interested in the heart, cardiomyocyte, and heart tissue, which is a tough muscle, and it's got lots and lots of extra cellular matrix connecting those cells. So I just worry that the expansion of these cells would be tougher.
Well, in the most popular expansion protocol, we apply fluorescent antibodies first. We anchor the fluorescent antibodies to the polymer, and then we use very strong proteases to destroy all the original sample. So that's the strongest way of doing it, and could be applied to things like heart, I would think. But the more recent versions that we have not yet published, where we expand proteins away from each other, we've mostly been testing that in brain. But I've definitely seen people expand things like muscle and heart. Although I'm trying to think of how much of that is. There's so many papers coming out on this. I'd have to check to see what the status is of it.
Thanks. I'll look it up.
Okay. Yeah, I'm chief tech support person, so just drop me a line if you have any questions.
Sure, I will actually. Okay.
Yeah, other questions?
Hi, James here.
Hi.
Hi. I did actually have two questions. I know you said only one, but if I quickly ask both of them, Ed can choose to answer one or the other. One is, has anything been done using these techniques to look at the behavior of the coronavirus in humans? And the other question is, what is it physically about the polyamide matrix that makes it expand so extraordinarily isotropically?
I think we probably have time for both questions.
Go ahead, sorry.
Yeah, I think we probably have time for both questions.
Thanks.
Yeah, so we ourselves are actually hopefully going to receive some brain tissue soon that we can look at and see if the coronavirus is hiding in the brain. And others have contacted me as well, seeking to apply expansion to try to figure out where viruses are in the body. Because of course, viruses are nanoscale. So yeah, that's exciting, and hopefully will yield some insight. So there are a couple of things, right? We weave this polyacrylate mesh very densely. The spacing between polymer threads is only a couple nanometers. So that's one reason why the expansion process is so isotropic. The second is we soften the tissue, as I mentioned earlier, using enzymes, detergents, and heat. So if the polymer is expanding with roughly, let's say, 2 nanometer spacing, and the endogenous tissue is not resisting the expansion, you might imagine that the fundamental resolution is around a couple nanometers, and if you can expand it without resistance from the tissue, then you might be able to approximate that. We're not there yet. We have a bioarchive preprint with a slightly different polymer, a somewhat different polymer composition, same acrylate strategy, but a different geometry of the monomer. and that allows us to get what we think is single-digit nanometer resolution, 9 nanometers, so heading in the right direction. But yeah, I'm excited that we might actually get down to single molecule resolution at some point in the future.
Thank you. That's impressive.
Professor Boyden.
Hi.
Hello, my name is Mark Oeller. Thank you very much. I actually worked on expansion myself a few years ago. I'm very excited to see the progress in it. And actually, I want to ask you about the thing you just mentioned. Do you think, what's the limit of expansion microscopy? Because you also published a paper about the iterative process of doing it more than once in a row to go actually to 20x. Do you think you can even go for even higher resolutions?
Oh, yeah. Yeah, so as I mentioned, yeah, we already think we can get down to 9 nanometer resolution. There are other preprints kicking around where people are claiming resolutions as good as 5 nanometers. Although we haven't yet gotten that in our hands. But yeah, it's exciting. I think there's sort of as I mentioned earlier, the space between polymer threads is probably around 1 to 2 nanometers from small angle X-ray scattering. So that's the size of a single biomolecule. So can we get down to single molecule resolution? I'm excited that in the future we might be able to.
Okay, thank you very much.
Hi, do we have any more questions for Prof. Boyden? Otherwise, I think we'll. Oh, yes.
I would ask a follow-up question if that's fine, if there's still time.
Go ahead. Please do.
Do you have any ambitions of trying to visualize dynamic processes with expansion microscopy? Because I mean, in a sense, obviously it's somewhat stationary, right? Because you digest the cell. But do you think it might be possible to image basically snapshots from certain processes if you go down to single molecule level?
Oh, potentially, sure, yeah. I mean, the bacteria expansion paper from Stanford that I mentioned earlier, they also tried expanding bacteria without softening the cell walls. And you can imagine making mechanical measurements of cell protein-protein or molecule-molecule interactions by letting them interact while pulling on them. Yeah, so I think there's a lot of exciting possible directions there where you have what you might call a quasi-living state. I mean, anything with diffusion will be eliminated because you've anchored all the molecules in place. But still, you might be able to study protein-protein interactions or mechanical forces or other things in this quasi-living state.
Very, very exciting. Thank you.
Yeah.
Are there any more questions?
Well, don't hesitate to check out our website and try it out, the protocol. We're always happy to answer questions because we really want to help people solve real biological and scientific mysteries. So yeah, stay in touch and always happy to help.
Maybe I'll just follow up with another question if that's all right. Regarding the light sheet microscopy technique which you mentioned, how exactly does it increase the speed? For instance, I quoted you mentioned 1 by 1,000 times. So what's the mechanism behind this increase in speed?
Well, it shines light from the side, and you can take a picture at a 90-degree angle. So each picture can take an image of like a 2D slice, and that works because the sample's transparent. That means you can go much, much faster than, for example, scanning a single laser point, right? Because you're taking a picture of a whole section.
I see. That's interesting.
Yeah, it's called light sheet microscopy. It's a whole field. But you don't need that microscope to go still at very appreciable speeds. And many people are doing expansion and then just imaging with regular old microscopes that they have access to.
All right, I think we will bring this lecture to a close. Thank you,
Great.
Professor Boyden, for this amazing talk. A recording for this lecture will be made available on our YouTube channel shortly, and thank you all for joining us today. Have a pleasant day ahead.
Have a great day.
Thank you very much.
Bye-bye.